and the third column is for ovale, and the fourth column is for falciparum. Okay, I will try to tell you some points regarding this. It might come in AIMS exam, so that's why I'm discussing this. Okay, so in the first row, in fact I should say the first column wise we'll discuss this. Okay, so you can see up in Plasmodium vivax the first one. What is that first one? That is the early trophozoite form, the ring form of the Plasmodium vivax. Okay, the second one is the late trophozoites with Schaffner's dot. Okay, and the important thing to be remembered here is note the enlarged RBC. Okay, and the third one is the late trophozoite with amyloid cytoplasm. Okay, and that is typical of Plasmodium vivax. The fourth one is the late trophozoite, again with the same stuff, that is amyloid cytoplasm. And the fifth one is a mature scisont with merozoites. Okay, as you can see these merozoites here with clumped pigment here. Fine. And the sixth one is a microgametocyte with dispersed chromatin. You can see up the chromatin which is dispersed up. So it's a microgametocyte which is dispersed chromatin. And the seventh one is a macrogametocyte with a compact chromatin. Okay. So as per the questions concerned, one thing is for sure that two things are there from this. The one is this amyloid cytoplasm. That is what is important for this. And another one is this, the size of the RBC is increased up along with Schaffner's dot. That is typical of Plasmodium vivax. Okay. As you can see in malaria also, the first one, number one, is the early trophozoite form, that is the ring form. Okay. The second one is the early trophozoite with thick cytoplasm. Fine. And the third one, this is what is being asked regularly here. So remember, the third one is the early trophozoite, and that to the band form. Okay. And the fourth one is the late trophozoite band form along with the heavy pigment. Okay, this is frequently asked question regarding malaria, the band forms. Fine. And the fifth, which you can see here, is a mature scisont with merozoites, as you have seen up. And other than that, the sixth one again a microgametocyte with dispersed chromatin. And the seventh one is another macrogametocyte with compact chromatin. So this all things, 7 points regarding the Plasmodium malaria. Okay. Now coming to the ovale, as you can see, the early trophozoite ring form and a slight dot is there, and that is what is known as James dot here. Okay. Besides this, in the second one you can see the early trophozoite, and again remember there is a size increase. So remember, vivax and ovale, they have a large RBC. Fine. Then the third one is a late trophozoite in the red blood cells with certain fimbriated edges, they call that. Okay. Then you have this fourth one. This is the developing scisont with irregular shaped RBCs. Okay. So you focus upon the numbers only. The fourth one we are talking in Plasmodium ovale, and the fourth one is the developing scisont with irregularly shaped RBC. Okay. And the fifth one here in the third column, the fifth one, okay, in the third column, the fifth one, the mature scisont with merozoites. You can see that, as in all others, this is also a mature scisont with merozoites, and they are arranged irregularly here. Okay. And the sixth one again is a microgametocyte with dispersed chromatin, and the seventh one is a macrogametocyte with compact chromatin. Okay. Now coming to the most important fellow, that is the Plasmodium falciparum. The first which you are seeing up, it is the early trophozoite form, and that is also termed as Ecol forms. Okay. And the second one is the early trophozoite. Fine. And you can see up, one ring is in the headphone configuration here, and other one is having a double chromatin dots, like that headphone one is like double chromatin dots. Fine. So you can see up one of them, this is having a headphone like configuration, and another name for that is double chromatin dots. Okay. The third one is the early trophozoite with Morus dot. Okay, as you can see here in falciparum we have Morus dot. Fine. And the fourth one is the late trophozoite with the larger ring and the Morus dot along with that. And the fifth one is there, that is the mature scisont with merozoites, and that is what is the most important stuff. The sixth one, a microgametocyte with dispersed chromatin, okay, and the macrogametocyte, the seventh one. And as you can see the shape of these fellows, shape is banana shaped gametocyte, okay, and the seventh one, that is what is a macrogametocyte with compact chromatin, okay. So these all are the various stages of the Plasmodium in various forms. The first one we have discussed is Vivax, the second one we discussed is Malari, and the third one we have discussed is Ovale, and the fourth one is Falciparum. And as you can see up, you should go column wise along with the required information. Listen to this video once again to relate up to the things to the best of your knowledge, okay. In all, if I want to summarize all this, I just want to allow you to focus upon 2-3 things very importantly. The first one is that the Plasmodium vivax, this particular fellow, is having a large RBC, and along with vivax the ovale is also having a large RBC. Fine. The vivax fellows, they have Schaffner's dot, and the ovale one is having the James dot, okay. And whereas in Falciparum this is what is termed as Morus dot, and in Malari this is what is Siemens dot. Fine. In Malari these particular forms are there which you are seeing up, that is the band forms. Another important question here. And in Falciparum, as you can see in the second one, that is the headphone like configuration or double chromatin sign, that what you need to remember. And the last one is there, that is the banana shaped gametocytes, that is the characteristic of Plasmodium falciparum, okay. So remember all these points, these are being asked again and again. Thank you so much.